Celprogen bases its excellent reputation for quality and product purity by adhering to a strict regimen in the lab. Here's the correct procedure for seating plated T75 and T25 flasks. Wipe down the fresh flask with 70% alcohol upon arrival. Transfer into tissue culture hood. Remove 100% media and transfer into 6 50 milliliter sterile conical tubes. Set aside for further processing. Note, in the T25 version of the protocol, there will be 2 50 milliliter conical tubes as opposed to the 6 for the T75 version. With the remaining attached cells in the T75 flask, wash with 3 milliliters 1x PBS solution. This is from Celprogen catalog number P1408-013. Note, in the T25 version of the protocol, wash with 2 milliliters of the 1x PBS solution. Incubate at 37 degrees centigrade at 5% CO2 for 2 hours with 10 milliliters of the complete growth media. Note, in the T25 version of the protocol, use 7 milliliters of the complete growth media. The reason we do this is to reduce shipping-induced stress on the cells. Remove and safely discard the 100% media. Wash flask with 3 milliliters of the 1x PBS solution for 2 to 3 minutes. Remove and properly dispose of the 1x PBS solution. Note, in the T25 version of the protocol, wash with 2 milliliters of the 1x PBS solution. Trypsinize the remaining cells with 3 milliliters of trypsin EDTA. That's from the Celprogen catalog number T1509-014. Note, in the T25 version of the protocol, use 2 milliliters of trypsin. Prior to observation, gently tap flask to dislodge adhered cells. Neutralize trypsin with 7 milliliters of fresh complete growth media with serum to bring total volume to approximately 10 milliliters. Note, in the T25 version, the quantities are 2 milliliters of trypsin to which you add 8 milliliters of the complete growth media to yield 10 milliliters total volume. Transfer into one sterile 15 milliliter conical tube. Centrifuge at 100 G for 7 minutes to obtain a soft cell pellet. Safely discard the 100% supernatant media. Reconstitute the cell pellet by gently tapping on the conical tube. Transfer the reconstituted cell pellet into a single conical tube. Reconstitute cells in 10 milliliters of the complete growth media. Note, in the T25 version of the protocol, use 7 milliliters of the complete growth media. Transfer 10 milliliters of the reconstituted cells into a pre-coated T75 flask. Note, in the T25 version of the protocol, transfer 7 milliliters of the reconstituted cells into a pre-coated T25 flask. Observe the cells under the microscope prior to placing in the incubator. Incubate at 37 degrees Celsius 5% CO2 in a humidified incubator. After 24 hours of plating, inspect the cells. Please note, if the attached cell density is low, combine with suspension cell pellet into a total volume of approximately 10 milliliters for T75 flask or 7 milliliters per T25 flask. Acquire the 6 conical tubes of suspension cells created in Part 1 and centrifuge for 7 minutes at 100 G. The T25 version will have 2 conical tubes. Safely discard the 100% supernatant media. Reconstitute the cells in 1.7 milliliters of complete growth media per 50 milliliter conical tube. Note, in the T25 version of the protocol, use 3.5 milliliters of the complete growth media. Combine all cell pellets in the 50 milliliter tubes into a single 50 milliliter conical tube to reach a total volume of approximately 10.2 milliliters. Note, in the T25 version of the protocol, the total volume will be approximately 7 milliliters. Transfer the reconstituted cells into a pre-coated T75 or T25 flask. Observe the cells under the microscope prior to placing in the incubator. Please note, if the suspension cell density is low, combine with the attached cell pellet into a total volume of approximately 10 milliliters per T75 flask or approximately 7 milliliters per T25 flask. Incubate at 37 degrees Celsius 5% CO2 in a humidified incubator. After 24 hours of plating, inspect the cells. In the T25 protocol, if the cell density is high, plate the cell pellet into 2 T25 flasks for a 1-to-2 split. This concludes the seeding protocol for plated T75 and T25 flasks. If you have any questions or suggestions, please contact Celprogen Customer Service at area code 310-542-8822 or email customerservice@celprogen.com.